I grew up in Alice, just north of, I think the region that's covered by your organization, and but also belonged to a science club, which helped me, very early develop an interest in science. And I always loved the chance to get to share my enthusiasm, my love of science, and the fun it could be, and the importance that it could have to young people. And so I will be glad to now. Whoops, if I can figure out how to do this properly. Just seem to be having some problems here. Here we go. Okay, here we go. Sorry for the fumbling. So what I want to do today is talk about some work that goes back to the 80s, actually, the 1980s, that I've been carrying out. It was really in basic immunology, but that has led to a way that of treating cancer in a really curative way for, I think, one of the first time, and hopefully will serve to not only educate you as to how this could work, but also how important doing basic science research is. I'm having some. Okay. I want to start off by just showing one patient that illustrates the success of this. This is a CAT scan of a patient. Her name was Sharon Belvin. This was in 2005. She was 22 years old, had just finished college, just gotten engaged to be married, and just about to start her career as a teacher when she was diagnosed with metastatic melanoma, meaning it had spread all over her body. And what you can see here, these dots are metastases of melanoma growth. It's a skin cancer, but it goes all over the body. And she had 31 of these in her lungs. And after treatment, they were all gone. Whoops. It's a different way here. And she also had a half-centimeter brain metastasis, which was even more troublesome. So she underwent chemotherapy, high-dose IL-2 therapy, and another immunotherapy, which doesn't work a lot of the time. But for this, she got radiotherapy. And if you see the movie Breakthrough, you'll see her with the headgear on and everything as she prepares for, radiotherapy in her brain. But that didn't work. But after she got anti-CTLA-4 treatment, that was completely resolved. That was in 2005. This is a photo she sent me in 2016, 11 years later, with her two children. She's gotten married, has 2 kids, and she's still doing fine in 2020. She's become a close friend, and she actually went with me to Stockholm for the Nobel Prize thing, and is really, really a courageous person who went through 2 years of just grueling treatments before she was finally cured. Anyway, so back to the scientific issue. Why would you want to use immunotherapy to treat cancer? Well, cancer is a disease of mutations, and something goes awry, you're exposed to carcinogens or whatever, and you start picking up mutations in your genome. And when you finally get one that affects a gene that regulates cell growth, for example, or something like that, that's normally off, except when a hormone or a growth factor binds to it and says, now you need to start dividing. You can have mutations that say, lock it on, like hot wiring the ignition switch. And those cells become cancer cells. And after that, their genome becomes very unstable, and they start to develop more mutations. And so cancer really is not just, you can't really talk just of lung cancer or breast cancer. I think it's many, many different diseases. And some people like to, divide it rather than tissue of origin with the gene of origin, but neither of those actually works particularly well. But what we learned through the war on cancer time, when The goal was to sequence the genomes of cancer cells and find the mutations that were different than normal and drove cancer, and developed enzymes, inhibitors that would stop those, block them. They would lead to remission, but because there were so many mutations, the disease would almost always recur within a few years, because in the population of cancer cells, there were many mutations with many different drivers there. But immunotherapy has several advantages. One is specificity. What immunotherapy detects is anything that's different about cells, that's not normal, that shouldn't be there. And so the products of these mutations are all targets for the immune system, regardless of whether they drive the cancer state or not. The second one is memory. Once you've got T cells, which is what we're talking about here, are the soldiers of the innate of the adaptive immune system. You've got them forever. They'll stay with you for the rest of your life. So if the cancer comes back, they'll more immediately, there'll be many of them, and they can attack. And finally, there's adaptability. You probably have 10 to the 15th power different T cells in your body. That's a huge number. And your immune system can change according to the insults that your body is suffering at any given time. So it can adapt to it. And other drugs can't. Once you give a chemotherapy agent, when it's gone, that's it. If it didn't kill every tumor cell, it's going to come back. Radiation, the same. Surgery, the same. If any cell escaped the surgery, it's going to grow back. But the immune system can change, just as it does to in flu viruses, or now we know coronaviruses and things, and protect you and provide you with memory. So so it's definitely a match for cancer, we think. And so immune checkpoint blockade is what I helped develop through my work. It was really a paradigm shift in cancer, because what it says is you could treat cancer not by targeting or trying to kill the cancer cells, and not by developing vaccines or anything like that to try to turn on the immune responses. But we learned was that there are many inhibitory pathways that exist to control the immune system and make sure it doesn't attack normal cells, that can suppress tumor responses. And so we developed a way to block those inhibitory pathways to unleash anti-tumor responses. And so we got there by understanding fundamental mechanisms of T cell activation and regulation, basic science. I started this work just to understand, because I was curious how T cells worked. When I was in college, I took a course, and the professor said, antibodies were well known, but one lecture at the end, he said, there are these new cells that have just been discovered that could go through your body and identify anything that's wrong and deal with them. And I said, how does that work? And he said, we don't have a clue. I don't even know if they really exist. But anyway, so at that time when I was in college, I said, well, that's what I'm going to devote my career to, is figuring this out, how that works. And so basically, to sum it up, here's some of the things that we learned. One is that T cells, just recognizing a foreign antigen through the T cell receptor, the structure of that I worked out and published in 1982 when I had a lab at Smithville, Texas, under MD Anderson, is necessary to recognize that foreign thing that the T cells see. But by the late 80s, we showed that that's not enough, actually. It's not just an on and off switch. There's another signal 2, it was called, or co-stimulatory signal, that we showed by the late 80s was a molecule called CD28. And if a T cell doesn't get both of those signals, it doesn't get turned on. Most tumors, they have antigens, which can give a signal through the antigen receptor, but they don't have that second signal. And so they remain invisible to the immune system until such point that tumor cells begin to die, and then a necrotic death, we think, because they get too big and they can't get enough oxygen or food or whatever. And these cells of the innate immune system, called phagocytes, dendritic cells, pick up bits of the tumor cells, redisplay them on their surface in the context of the two molecules, the B7 molecules, that bind to CD28. And those two signals then, it's like the ignition switch and the gas pedal, tell T cells to start growing. Except we discovered later on that there's another molecule called CTLA4 that's turned on when you start that process, and it takes it a while to build up, but it accumulates, and after a while it can just turn off those other signals and stop an immune response. And so in the mid-90s, I had the idea, well, let's just block this, disable, it's the brakes, so let's just disable the brakes with an antibody to CTLA4 and let T cells keep running as long as they can, because CTLA4 comes up to stop an immune response to help keep it from hurting you. It's a race. If the tumor cell grows fast enough where it can kill you before the T cells can catch up, it wins. But if you could unleash the T cells to just keep going long enough, then I thought that maybe that would work. And so we developed an antibody to CTLA4 and showed that it could actually lead to tumor rejection by letting T cells keep growing. And there were a number of things about this that were really compelling as a cancer therapy. One was you could take advantage and use it by itself to take advantage of necrotic cell death that happens when a tumor gets too big and starts causing damage. Or you could alert it by putting vaccines there, which would be presented on these cells immediately. Or you can kill tumor cells with conventional things. And if you did that in combination with anti-CTLA4, with blocking that pathway, you could give chemotherapy, radiation, the properties of immunotherapy, memory. So after the drug's gone, it doesn't matter. You've killed some cells and you've gotten T cells. You give it adaptability because from then on, the immune system can handle the tumor. But also the fact was that since we're treating the immune system and not the tumor cell, this has the possibility of working against any kind of cancer. Anyway, this is one of our first mouse experiments. We gave mouse tumors, and if we didn't give them any antibody, they would grow and kill the mouse. If we injected antibodies that blocked that second signal, the tumors actually grew faster, as you can see here, and indicating that the immune system was trying to do something, but just couldn't get it done. But if we injected anti-CTLA4 antibodies, it took a while for it to get started, but the tumor would then be rejected by the T cells. And I won't show you all the qualitative work, but there's many more T cells attacking the tumors. And those mice, we could challenge them for the rest of their lives without treatment, and they would be totally immune to re-challenge with that tumor. So we did this with many different kinds of tumors. And after a time, found a company called Metarex that had developed a mouse, very cool mouse, where they had replaced all of the mouse immunoglobulin genes with human, because you cannot inject a mouse antibody into humans because it will be treated as foreign and will be eliminated. So you have to have a human antibody. So they have these mice that would make a human antibody to human CTLA4. And later on, Bristol Myers Squibb, a large pharma company, joined us. Anyway, this went into trials after about 2001 in many tumor types, small trials. And just as we predicted, it wasn't tumor specific. Melanoma, prostate, kidney, bladder, ovarian, lung, many other kinds of cancer responded. Now, we never saw any adverse events in mice or in the safety studies that we had to do in cynomolgus monkeys. But in humans, we do see a lot of adverse events. Most of these are just inflammatory things that can be treated satisfactorily and you still get tumor rejection. But these days, we know after hundreds of thousands of people have been treated that there are some, in a very small population of people, cases of inducing autoimmune diabetes, and heart attacks and other things that we're working on. And that's a separate talk. But anyway, but generally it's well tolerated, much better tolerated than most chemotherapies, and can be treated without affecting the anti-tumor response. So this went into the clinical trial that was started about 1997 or so, finished in 2010. That produced these data. This is a complicated figure, but this is the typical curve that you use to evaluate cancer patients. You start with a large group of patients. In this case, it was over 800, and you divide them into a treatment group, and in this case, a placebo group. This was melanoma, and it was melanoma because of a couple of reasons. One, we had noticed, people, we meaning the field, that melanoma was highly populated with T cells. And so it was alerting the immune system, but for some reason they weren't working. And so it was a good target for that reason. But the other one was no drug ever of any type had ever prolonged survival in a proper randomized phase 3 trial. So there was a placebo control, which basically had no hope of treatment, but what it showed was an improvement with N-acetyl-A4 of about 4 months, with a median survival of about 7 months after diagnosis. And this was the typical of the time in the control arm, that once you were diagnosed with melanoma, 50% of the people would be dead in 7 years, and no drug had ever prolonged that, as I said. So there was a very sad and lethal drug. But you can see here, treatment with N-acetyl-A4, in this case, the antibody ipilimumab that we made, moved it over by about 4 months. But the coolest thing, though, was if you look down here, after about 2 to 3 years, the survival curve flattened out. And after about 3 years, nobody, or very few people, died. So there was this tail on the survival curve. And I'll come back to that later, which nobody had ever seen before with any chemotherapeutic or any genomically targeted drug. But why is it just, anyway, this was followed up in 2014 with a study of 5,000 patients, which by then there was 10-year data available for. And that data actually shows that after about 3 years, the curve starts flattening out. And at the end of 10 years, a little over 20% of patients are still alive. And that means by any definition of treating, particularly this type of cancer, they're cured. A curative effect, one in 5 patients, which was good, obviously, because this was a lethal disease, pretty much uniformly lethal. But the question is, why not everybody? It's disappointing in that way. Why not everybody? And there are a number of possibilities. One is that this works, as I showed you, during priming of T cells. And so if that's not going on when the drug's on board the patients, it's not going to help them. And other ones. But obviously the more interesting one is maybe there are other checkpoints, cell intrinsic checkpoints that limit immune responses because it has to be controlled. And sure enough, that turned out to be the case, because in 2001, just as our clinical data was beginning to come out, it was shown that a molecule called PD-1, discovered by Tasuku Hanjo, with whom I shared the Nobel Prize, was another checkpoint that on T cells that could suppress them. It's a little different than CTLA-4, however, because it works on fully functional T cells, because the targets, the tumor cells themselves, if a T cell approaches a tumor cell, starts making a cytokine called gamma interferon that will kill a tumor cell, the tumor responds by upregulating one of the ligands for PD-1, PD-L1, and it actually inhibits the activity of fully formed T cells. So it works at a very different stage, but it's another checkpoint. The immune system is very complicated, very powerful, and so there are multiple checkpoints built into the system. to allow regulation of it. But an antibody was made, it very quickly went into critical trials. And in 2012, there was one reported where it showed very good. Now, I will define complete response means all of the tumor went away from the patients. Partial means some fraction, usually more than 30% or so of the tumor went away. And so those are called objective responses, and that was about 28%. 6% or so of the tumors just stopped growing. And non-small cell lung cancer is about 18%, really good. And renal cell cancer, also very good. In colorectal cancer, there were no responses, although in an expanded trial, there were some responses. And as I'll mention briefly later, that was because there's a subset of cancers that have defects in DNA repair. So they generate more mutations, more mutational products for the immune system to see, and they do respond. It's about 15% or so of colorectal cancer. But there were no responses in prostate cancer. As I said to you, with CTLA-4, there were responses, and we figured that out, and I will tell you about that, why that is, and I'll tell you about that in a few minutes. So where do we go from here? So we got two agents, which are mechanistically different. I don't have time to talk to you about it, but we now know at the molecular level why they're different. And it's very interesting and gives us clues as to how to combine them, and also with other things intelligently. But anyway, this time we had two agents that looked like they worked differently, so why not put them together? So this is a trial designed by my friend Jed Walchuk that I worked with a lot when I was at Sloan Kettering. But this is what you get with anti-CTLA-4, again, about a 22% flattening of the curve. With the combination of that with the PD-1 antibodies, it goes up to close to 60% actually. And this just follows them out for about a year and a half. But we now have 5-year follow-up data, and it stays above 50%. And there's no reason to think that it's because anti-CTLA-4 is involved. There's no reason to think that that won't go on to 10, 20, whatever years and be considered curative. And so we've taken this one disease, melanoma, which was pretty much uniformly lethal. And if you were diagnosed with it before 2011, you were going to be dead with a median survival of 7 months of diagnosis because no treatment worked, to being at a point where over half of the patients can look forward to a response that lasts a decade or even more. And so, but why just half? We're still not there. But nonetheless, the FDA approved CTLA-4 in 2011, PD-1 in 2014, and there are now 6 or so different PD-1 antibodies. But this just shows all the cancers for which there are FDA approvals. Now, what FDA approval means is that randomized clinical trials show that these drugs are better than any of the other treatments that are out there, which is important. But moreover, if they're FDA approved, then insurance companies pretty much have to pay for them, which for patients is the more important thing. But it's not just melanoma anymore. It's lung, kidney cell cancer, colorectal cancer, as I mentioned, for a subset, head and neck cancer, some blood cell cancers, liver cancer, Merkel cell carcinoma, which is an even more lethal kind of skin cancer, which kills people in a much shorter period of time, of which there was no treatment whatsoever. Again, stomach cancer, cervical cancer, breast cancer, skin carcinomas. Anyway, it just goes on and on. Most interesting, last year, the FDA approved this drug, because in a minute I'll talk about the role of mutations in it, for any kind of cancer, regardless of the kind of tissue it arises in, that has defects in DNA damage repair and is likely to have more antigens. And so a really forward-looking kind of therapy that shows where we're headed. And these are happening right now, every month or so. approval rate is going on and up. But we still have a lot of work to do to get the response rates higher. Instead of, we're about 30% now in kidney cancer, for example. So how do we get that higher? How do we get melanoma to 100%? How do we get it much higher? And there's certain cancers, glioblastoma and pancreatic cancer, which, has taken many people that we know, including Ruth Bader Ginsburg, most recently, very lethal cancer that we've got some hints, but no real treatment. So we need to keep working to figure out how to make it work there. And so Pam Sharma, who is my partner, who's a physician scientist who works in genitourinary cancers, bladder, kidney, and prostate. And I established at MD Anderson institution we call the immunotherapy platform. And our job is to carry out clinical trials based on what we understand about these checkpoint blockade, and then take samples from the patients, from their tumors, from their blood, and take them back to the laboratory and see what's going on, what happened. What happened to the cells of the immune system? What happened to the cells of the tumor? What happened in general, so that we know. We know now from the successes, we know what a good response looks like. So we need to know, if we give a drug and it only works partially or it doesn't work at all, what was it that it lacked? Because the immune system is complicated. There's not only T cells, but they're macrophages, and they can be suppressive or helpful. There's other cells of the innate immune system. It's a very complicated process. And I will just tell you that because of the success of this, there are drug companies, there are 3,000 trials underway now by drug companies trying to find something to combine with PD-1 blockade agents in particular to make it better. But all they do is they look for, does it work clinically or not? And if it doesn't work, they just move on to the next thing, when they don't do the scientific work to understand what happened. Maybe it wasn't a home run, but maybe it was a second base hit, And so we decided to do, reverse translation, instead of just concentrating going from, laboratory to bedside, to go from, the clinic back to the laboratory and analyze things and see if we can't figure out answers to those questions. And so this is the thing that we did, and perhaps a little esoteric, but what we did was first of all, we got permission, because we can't take specimens from people without their permission. But we have permission to ask patients that have any kind of cancer, under any kind of treatment, to give us specimens of their cancer that we can analyze in the laboratory. And we're involved in 113, actually it's 117 now, different clinical trials. We've enrolled more than 4,000 patients and gotten a lot of samples to analyze. And I sort of concentrate now for the rest of the talk on what we've done in one particular kind of cancer, and that's prostate cancer, which, as I showed you, there were responses with anti-CTLA-4, but not with anti-PD-1. So why is that? What's the basis for that, response? Because not very many responses were observed with anti-CTLA-4. In fact, a phase 3 randomized clinical trial of anti-CTLA-4 was carried out, and it failed, because even though there were some responders, it didn't meet the level of statistical significance. And this was CTLA-4 only. So again, we decided to figure, why is that? Because there are some patients that responded. So there are a couple of things about prostate that are interesting. One of them is that from all the cancers that have been looked at so far, we know from the work of many very dedicated genomic scientists that different cancers have different rates of mutation. Cancers like melanoma have a lot of mutations, hundreds to maybe even thousands of mutations per cell, because they're caused by mutations induced by radiation to ultraviolet rays, to sunburn. And people go out and they get, blistering sunburns. And so they get an intense exposure to carcinogens at one time, and they end up with thousands of mutations in skin cells. And some of those end up causing cancer. Lung cancer, various types. Stomach is because of, it's been shown to be because of carcinogens in the things that are generated when you burn tobacco and smoke it into your lungs. But those metabolites of those things also go to the stomach and to the throat and to the bladder and so they're very high rates of cancer, and they're associated with very high rates of mutations by those carcinogens. Those cancers in these red boxes have been shown to respond to treatment with monotherapy, that is either anti-CTLA-4 or PD-1, as I told you, because the immune system loves mutations. That's what it likes. Anything that's different, we need a tumor cell and normal cells it's going to go after. And that's why those responses are so great. But when you get over here, the response level is lower. It's not very high. And kidney cancer, for example, has a rate that you might not expect it to respond at, but it does respond again very well. And Dr. Sharma led 2 trials in kidney cancer that led to approval of the monotherapies there. And there are other issues about kidney cancer other than just mutational rate I don't have time to go into that make it respond. But also you see here's pancreatic cancer, for example. It doesn't respond very well. It's very lethal, and we haven't had much success with. But down here is prostate. It's even lower mutation rate than pancreatic, and yet there were some responses. So what's that all about? Why is it so low? And why does it respond to anti-CTLA-4, but not PD-1? So one of the things that we did was to ask why those patients that got anti-CTLA-4 were responding. Again, it has a very low mutational rate, ranging to, instead of hundreds to thousands, maybe a dozen per cell. And so what we did was we conducted a trial, actually Pam designed it, where we took patients with metastatic prostate cancer, but we got samples of their tumor. They were taken out, and we did genome sequencing, and then also sequenced RNA to know which of those genes were expressed, what the mutations were from the DNA sequencing, and which ones were actually being expressed at the protein level, at the RNA level at least, from RNA. And then use those to think about what might have been antigens. So the numbers were low enough where we just made peptides to all of the expressed RNA products that we observed in these patients, which were different from every patient to every patient. And then the patients got 4 doses of anti-CTLA-4, but at each point we took blood samples from them and looked if we could find T cell responses to those, as well as common prostate cancer antigens. And so we had some patients, these with the blue lines with the green heads, that actually responded, meaning more than 6 months of treatment, where most of the patients did not respond did not survive that long. But the ones that responded that long after being treated, again, all these patients were treated. And so then all of the patients, those who didn't respond or not, we had their sequences, their genomes, and we knew how many mutations they were that generated new amino acids and how many were expressed. And I'm here going to talk about patients 5 and 7, both of you responded. And they had 12 and 10 mutations, which were relevant totally. I mean, again, we're talking in lung cancer and melanoma, hundreds to thousands. This is, barely a dozen mutations. And so we could make the peptides and analyze the responses. And so this shows patient 7, who was a responder, for example. This is some control responses. This is measuring the number of T cells that proliferate and make cytokines. Anyway, responded to this mutation, but not to the wild type version of that gene. Had a second mutation. That was to a mutation in the guanosine nucleotide exchange factor. This other one was in dihydropyrimidine. It doesn't matter what they were, really. They were just proteins that were made in the cell and had mutations, and the immune system puts those on the cell surface and says, is this different? Anyway, these were single amino acid changes, and in these patients, T cells could respond to the one with the new amino acid and not the wild type. And so that was very interesting that they could respond. This is the second patient who only had one response to another protein called protein L isoaspartase O methyltransferase. Again, one amino acid change, and you got T cells, and against the parental, you did not. And so anyway, these patients responded. And so the interesting thing about these patients, though, is they also made responses not just to those antigens, but those neoantigens, but also to antigens, prostatic acid phosphatase, which is essentially found only in prostate cancers, and prostate-specific membrane antigen. So many of those, but PSA, I'm sure everybody's heard about. There were T cells that in many patients, but they didn't necessarily respond. So we don't know definitively whether these responses were due to those mutations, but are these more shared antibodies? But it shows that you can get responses in prostate cancer despite the low mutational rate to immunotherapy. But it's been thought for a long time that maybe T cells can't get into prostate. And so the idea here is a paper that Dr. Shorman and I published. There are basically two types of tumors: those that are heavily infiltrated by immune cells, like melanoma, and all you have to do is release the brakes that are provided by the checkpoints, and you're there. But there are other tumors, as I told you, such as prostate, that have and pancreatic cancer, that have very low mutation rates, but also they are very cold. When you look at untreated patients, their deserts, are cold, depending on how you look at them. They don't have any immune cells in them. So you have to get something to do to get the immune cells in. And so that's something that we looked at. Dr. Shorman designed a trial where we took patients that had localized disease and were going to surgery, first a conventional therapy with hormones, ablating testosterone, which can drive proliferation of cancer or not, but then, getting N-acetyl-A4. Then we went to surgery and we got the entire tissue that we could analyze, for the presence of immune cells. And so just to quickly show you the data, these on the left, this is an antibody, these are cells. So this is immunohistochemistry. These are pieces of tumor that we're looking at in the microscope that have been stained with antibodies to CTLA4, which would give you a brown stain. But if you look here with CD4 kind of T cell, CD8 another kind of T cell, FOXP3 another kind of T cell, CD45R0, which would pick up all T cells, ICOS, which would pick up the T cells that we know are induced by N-acetyl-A4, granzyme B, which would pick up T cells that are actually capable of killing T cells, then macrophages, which would, perhaps these would indicate that they're cells that could present antigen, but they're very cold before they get the CTLA4, nothing going on much at all. But after treatment, you can see all these brown cells with CD4, CD8, some with FOXP3, a lot of T cells in general. It's ICOS molecule, cells making granzyme ready to kill. So completely changed it. So this looks more like melanoma than it does like prostate. just by giving anti-CTLA-4. So it's clearly driven T cells in. But what happened was when you treat, we did RNA analysis just across the board to ask what kind of genes would be expressed of molecules that were positive or inhibitory to T cell responses. What we found is that after, this is a fold increase, that after treatment would induce PD-L1, VISTA, and other molecules, LAG3, all of these molecules, which are inhibitory molecules after CTLA-4. So it drove T cells in, but resulted expression of these inhibitory antigens like PD-1, for example, on the T cells. You can see here, nothing before treatment, lots after treatment. PD-L1, the molecule that binds to PD-1, nothing before treatment. Afterwards, on the tumor cells, tons of it. And a new molecule that had just been discovered, VISTA, it's an inhibitory molecule, also induced on cells. So we had gotten T cells in, but that made them put up an additional family of inhibitory molecules. And so this just shows the distribution now. Pre-treatment is the black dots, red are the after treatment. So the T cells themselves are expressing this inhibitory molecule, macrophages are expressing it, and the tumor cells are expressing it. So, we've gotten T cells in, but we've created a suppressive environment. And so we took these data because at that point, because of what I told you earlier, Mr. Myers-Squibb was enthusiastic about treating treating prostate cancer with CTLA-4 initially because the responses with it, but the phase 3 trial failed. And then PD-1 didn't show anything. So they just shut down their entire prostate cancer program. And we took these data to them and said, wait a minute, you need to give anti-CTLA-4 to get the T cells in, and then PD-1 antibodies to get rid of the inhibitory pathways that's been induced. And so this is the trial. You don't need to look at it very carefully, but basically it involves treating the patients with both of those antibodies to accomplish what I just told you. And so here's one patient from the first trial. It's PSA, which is a molecule that prostate cancer tumor cells make, went from 50 or so. The baseline is about 10, actually. But anyway, up to close to 500, and dropped after he got the first dose. Dropped even more to undetectable. All of the metastases, this patient was a complete responder. Everything went away. Anyway, one of the problems with this trial was because of the 4 doses, it's fairly toxic, and guys with metastatic prostate cancer are relatively old. It was too toxic. And so anyway, we have an expanded trial now of hundreds. This was a small trial of hundreds of patients with several different schedules of anti-CTLA-4 to try to overcome the toxicity. And so far, it's looking pretty good. What can I say? We don't have the data yet. But anyway, I show that as an example to what you can do if you continue to do science and look at the tissues of the patients that are being treated and learning what they tell you about how far it's gone. Have they gotten new T cells? Have they induced negative molecules or cells? We need to know. So the future, I think, that immunotherapy is going to be a part of all cancer therapies. Some of it's going to be blocking multiple checkpoints, as I've shown you. I didn't have time to tell you about one where we could give agonistic signal through 2 different positive therapies and have an effect. But anyway, that's going on too. But in combining those with conventional therapies, because unique, there are now 4 pillars accepted for treatment of cancer. The 3 classics were surgery, chemotherapy, radiation therapy. And then now, finally, immunotherapy is accepted as the 4th. But the nice thing about immunotherapy is it can play nicely because of the way it works with any of the others. And also there's strategies now for developing vaccines and taking advantage of our ability to block these checkpoints that would limit the responses to vaccines to make them more powerful, and in some cases maybe even treat precancerous settings. But the bottom line is I've just talked about the T cell receptor, CTLA-4, and PD-1. I mentioned briefly LAG-3, but there's lots of other molecules out there. And so you guys that hopefully are the aspiring scientists of tomorrow, there's plenty yet to do. Discovery science to find new molecules and how they work, but also, you know, how they contribute to science, to how they can be manipulated to contribute to anti-tumor responses. And so just to close, before I take questions, I'm sorry I've already spoken longer than I should have, but this is what, you know, oncology research consisted of before 2011, which was you took 1,000, maybe, anyway, large numbers of patients, you treated them with a new agent and try to see if you could extend the median survival statistically in some of those patients by a few months. But for the first time with CTLA-4, we did that, as I said, for 4 months. That would have gotten it approved compared to what was going on at the time. But the cool thing, again, was the tail of the survival curve flattened out. Unfortunately, it was about 20%. And this next one shows, it's not data, it's an aspirational curve, you know, which is why it wiggles here. That, you know, what we need to be working on now is just not extending the median survival of patients, but what combinations do we need of conventional therapies plus immunotherapies? Which molecules do we need to knock out on the inhibitory molecules on the T cells or on macrophages or on other cells of the immune system to get out of the way and get us closer and closer to 100%, and do it in as many kinds of cancer as we can. And, you know, it's a big challenge. And the answer is going to come through science. It's going to come through intense investigation and you know, smart applications to cancer treatment. And I think we can get there. I'm not sure we'll do it with all cancers, but I think we can do a lot better. I think we can get melanoma to closer to 100%. And I think that we can get kidney up to, you know, higher than 30%, which it is now. Anyway, on that, I think I will end my presentation, and I'll be willing to figure out how to do this and take questions. Anybody still has the energy. Thank you very much for your patience.
Thank you for your presentation. We'll begin asking questions from the document now.
So to start us off, the first question that we received were, what obstacles remain for the efficacy of the immune checkpoint therapy in non-melanoma related cancers? And I know you went over some of, in terms of mutations, how it relates to the current therapies. But I think this question is more going towards the forward sense in long term.
Yeah. Well, I think it comes from really, as I was trying to point out, you know, looking at drugs that don't necessarily give you a clinical signal, but what did they do? You know, we know, for example, there's a kind of macrophage, which is a part of what's called the innate part of the immune system that can turn T cells off. But we know they can be transformed under the right conditions to cells that can help T cells by presenting antigen. And, you know, if you give a drug and it gets you more T cells, but not enough, you know, it may be that you've got more of those inhibitory macrophages. So how do we inhibit those macrophages? You know, strategies for inhibiting macrophages. You know, it's basically just detailed science. It's hard work at understanding mechanisms and, you know, what drugs we can bring to bear.
Yep, I definitely think that answers that question. Next question, I think Bipul will be asking.
Yes, so our second question was, can immunotherapy begin from genome editing, and how can genetic engineering help with this?
Well, the genome editing has helped so far. in a different approach to immunotherapy, which is CAR T cells, where you retarget T cells by giving them genetically. You put in a gene that can make all your T cells go. You take T cells out of people, you put a gene into them that makes all of them recognize the tumor antigen, and you put them back in. That's what you hear about in leukemia a lot. The CD19, CD20. That's where it's at now, mostly leukemias. But there's still the PD-1 pathway, for example, is still an issue there. So people have gone and just cut the PD-1 gene out of those cells, which gives them longer half-life and keeps them from being inhibited. And it also allows you to put in, positive things if you want to. So there's a lot of opportunity now that's just, so far it's just getting rid of the bad actors rather than putting the good actors in. That's been done, but both are possible in that setting.
Thank you. We have a few more questions.
Sure.
How are the behaviors of naive T cells different from others?
Sorry, could you repeat that? Why are the differences?
The question was, how are the behaviors of naive T cells different from others?
Okay. Well, naive T cells, first of all, they're not doing much. They have essentially no cytoplasm. They're very small cells. They're just metabolizing enough just to stay alive and cruising around until they get a signal to the T cell receptor. And then it's an amazing change and it happens really fast. But it takes, as I said, it takes both signals from T cell receptor and CD28, that we showed in the late 80s. And then they just, they go crazy. They start dividing like bacteria, every 8 hours or so. So you go from, depending on the T cell receptor and the frequency of it, but they go from maybe a few dozen in your whole body to 1 million cells in 5 days or so, which you have to do if you're going to get rid of a flu infection, for example. But also, not only are more there, and now they can build cells, or they can make the cytokines that they need. Before they couldn't do it. Things weren't accessible. They couldn't be turned on. But also, the requirements for activation become much less. They're much more sensitive. They're on a hair trigger. So they differ in a lot of really profound ways from naive T cells. But that's the issue. You've got probably, I don't know, nobody knows the real answer, but maybe 100 million, maybe 1,000 million different T cells in your body for different things. Again, the repertoire to the species is like 10 to the 15th, and no person can have but a tiny fraction of that repertoire. So your repertoire depends on what protects you, but the repertoire available to the species protects the species. It's Darwinian. It's the way the system works. But if you have some that can react, there are not very many of them by definition, but then they expand very rapidly. They're going to generate an army. Few recruits is the way to get it. So memory T cell is just a few, and then they get activated, and then you have an army of cells that are also armed and trained to go out and kill.
All right, that was a very good explanation in relation to that question. And for our next question, it is how many protein receptors similar to CTLA-4 are known, and how can we make use of them too? So I think this is more generalized to other lymphocyte antigens that we can make therapies such as epilimumab from.
Yeah, very good question. There are probably about, I would say 300 or so genes from what we know that have some structural similarity. But when you start dissecting it down, it's probably a few dozen, that will have all the future features that are necessary. And they're not all equally important. Um, it seems that, CTLA-4 and PD-1 are the big ones. They are centrally involved in all responses. But you have to remember that T cells do a lot of different stuff. They go to different tissues, they make different cytokines, and all of those have to be positively and negatively regulated. And so, there are a lot, but I think the future is along those lines is for cancer. What we want to do is we want to get as many. We want to block the ones that limit proliferation of activity, get them out of there so we can get as active as possible. But for autoimmune disease, that's not what you want to do. What you want to do is find the ones that will actually turn off, certain cytokines, turn off T cells and develop different ways of inhibiting them. And so many of these are going to be useful, perhaps not in cancer, but other atherosclerosis, obviously diabetes, rheumatoid arthritis, and things like that. That may not be so useful in cancer. So it's the field is expanding way outside of just cancer treatment.
So our next question is, what is the difference between PD-1 and PD-L1, and which one is better?
Well, PD-1 is on the T cell, and it's the thing that inhibits T cells. PD-L1 is the molecule that stimulates it. And so PD-L1 typically is found, on the tumor cell or on macrophages. And still exactly what it does is kind of curious, because it's a little surprising that its expression on macrophages would actually play a role in cancers normally. Anyway, I won't get into details. But PD-L1, is quite important for that pathway, but it gives the signal. And all activated T cells have PD-1, but it doesn't. Its signaling again is quite complex, and it does not necessarily inhibit T cells until they reach a certain stage of differentiation that we're still working on, where the T cells express not just PD-1, but also a couple of other molecules. Only then does it become inhibitory. But I don't know which is more important, actually. Probably PD-1 in treating cancer. Certainly, there are tumors that a lot has been made, and you'll read about it, say that they type tumors for especially PD-L1 in making decisions to whether to treat them. And so they base that, on measuring by getting tumor samples and measuring PD-L1, except that we know that PD-L1 is induced by gamma interferon made by T cells. And so that doesn't necessarily just tell you the importance of PD-L1, it tells you that you've already got T cells in those patients, so they wouldn't have PD-L1. So it's kind of a circular argument, because if you give CTLA-4, there's PD-L1 everywhere, and measuring it doesn't help you with anything. And so I would say it's a draw. In some situations, it might be useful to measure PD-L1, others not. But PD-1's on every activated T cell. But it depends again on their phenotype. Are they at the stage of differentiation where they express these other two molecules that make them more susceptible to inhibition? I know that's a confusing answer, but that's the best I can do.
All right, so then our next question would be, what does the time between the treatment and the effect of CTLA signify? Is that the body is taking that time to adapt, or is it the response to any treatment time frame similar to that of the patient?
No, that's an excellent question. Again, as I told you, CTLA-4 works during priming. And so, you have naive T cells, And by blocking CTLA-4, you get more of them and they expand more quickly. PD-1 works later when you've already got T cells that have been shut down functionally. But they already know what to do. There's many, many more of them. And so it's been shown that when you give CTLA-4, you get new T cells. When you get PD-1, you just awaken the old ones, they expand. And so CTLA-4 actually, by its very nature, is slower. It could take months to work. PD-1 blockade usually works in weeks because of the very nature of where in the developmental cycle of effector T cells it works. And that makes a big difference, and when you think about dosing schedules and, how to give the drugs. But they're very different. You have to recruit the army and train them with CTLA-4. With PD-1, you just have to get them out of their nap.
That's a very illustrative way of answering that question. And I think Alvaro will be asking the next.
Okay, so the next question is, how is T cell differentiation in the thymus different from T cell differentiation in the bone marrow?
Well, good question. I would change it a little bit though. T cells, in the bone marrow, T cells, I should say, back up and say lymphocytes differentiate the bone marrow, and they make a lineage decision as to whether they're going to become B cells or T cells from the same precursor cells, actually. But in the thymus, the T cells are educated as to what antigens they're going to not see, because you need to get rid of T cells because the receptors are generated randomly, which is another cool thing that I worked on for many years. Because they're generated randomly. And so, like 90% of them, 90% of them are probably useless, but because they can't bind to the structure that presents antigens. But a lot of them are going to be reactive to self-antigens. So you have to get rid of the ones that react with self-antigens. So there's both a positive selection and a negative selection of T cells to give you a useful repertoire that comes out. So thymic selection for T cells tells them only whether they're going to be CD4 cells and do what they do, which is, a lot of different things, making cytokines, helping CD8s, or CD8s, which primarily kill, it's oversimplification, but they make granzymes, they punch holes in targets and inject stuff that kills them. But in CD4s, after they come out of the thymus, all they know is that they're a CD4. And so what they encounter and under the conditions tells them what kind of T cell they're going to be. Are they going to be a T cell that makes type 2 cytokines that mostly helps you make antibodies, or a Th17 kind of T cell that causes inflammation, or a Th1 type that kills virus-infected cells or tumor cells? So it's the flavor and the function of T cells that's affected once they come out of the thymus. It's very important. And we think actually, in some work that we published, that you can affect the kind of T cell that they become by messing with influencing the co-stimulatory antigens, CTLA-4, for example. Tends to, if you block that and ends up giving them a signal strength or whatever, that makes them become a Th1 rather than those other types of cells. That's the difference. In the bone marrow, all you get is B cell versus T cell. And in the thymus, it tells you the kind of T cell, CD8 versus CD4. And then for CD4s, it's after they come out of the thymus. When they first encounter antigen, there's a further differentiation step. It tells them the kind of CD4 T cell to become.
Thank you. That really helped sort out differentiation. So David will ask the next question.
Okay, so this question comes from someone who says his grandfather has prostate cancer and was instructed by the doctor to use Keytruda, but afterwards he got interstitial lung disease. So the doctor is now recommending PD-L1. Do you think he should continue with PD-L1?
You know, I hate to give individual advice because I don't know the total picture. So I really would. First of all, I'm not a physician. I'm a scientist, and I'd rather not answer that. If the best thing would be to go get a second opinion from somewhere else, from a cancer center, for example, to find out.
Thank you. So our next question is, can immune checkpoint therapies be effective in other autoimmune diseases, i.e. non-cancer diseases?
Yeah, that's a good question. You know, when we were doing our work in the late 80s and early 90s, I hate to admit it, but what we did was we tried to find things that would make autoimmune disease in mouse models worse, because we figured that would mean that you'd get a better tumor response. And we tried some approaches to try to shut off a response, but nothing that we did really worked. It's more complicated. But I think you ought to do the opposite. You know, if you could if you could make CTLA-4 signal better rather than blocking it, that might be a way. But we tried several approaches that we couldn't figure out how to do it. That's not to say it can't be done. I think it can be, but it's just it's going to take, you know, turning it around and trying to do the other way.
Yep, so I think this might be our final question. So what newer developments in immunotherapy interest you as a scientist?
Several. I hate to have to list them, but I think that we learn them right now. We're learning them, and they're different for different diseases, and we're learning them through what I described earlier, this reverse translational studies that we're doing through our immunotherapy platform, where we get samples from patients and look at what's going on, and we treat them with different things, and you know, stuff that's approved. We're not just going in and throwing stuff at patients, you know, and it's all done with consent. But you know, and I'm not sure there's going to be another big breakthrough in this area. I think we're just going to refine it and make it better. The question I get asked a lot is, what's the next big breakthrough? And my only answer to that is, if I knew it, I'd be doing it. You know, so I don't really know right now. I mean, that's where new ideas came from. You know, CTLA-4 came out of nowhere, and nobody believed it would work, and that's what took 15 years of, you know, hard work to, you know, get it into the clinic. And so, you know, right now everybody's looking for variations of things, but that's the way science goes. You know, you have a new discovery, everybody's against it at first, and everybody accepts it, and then you start working around it and making it better, and then in a little while somebody will come up with something absolutely new, and it'll probably be resisted for a while. But you know, but right now it's just, again, like I said, studying individual, you know, tumors treated with different things, including conventional agents, and seeing what happens, and how do you, in a scientific, informed way, decide, well, I'm going to give this to enhance T cells, I'm going to give this to enhance this kind of macrophage, this to inhibit this inhibitory kind of macrophage, etc. And we're to find combinations of drugs. There's a trial that's going on that we're involved with. I didn't design it, but in pancreatic cancer, where people are getting chemotherapy, radiotherapy, and then 3 different immunotherapies at the same time. They're getting five treatments, and it's showing a little bit of response, and that's what it's going to take, is figuring out the ones you can combine without causing toxicity. That's the next frontier.
Well, thank you for sharing your outlook. And I think David's going to close out.
Okay.
Thank you, Ms. Dr. Allison. That was a really great presentation. Thanks for all the insight too. At this moment, if everybody wants to take a screenshot, like for a photo of this, like you can turn on your camera.
Yeah, that'd be fine. Fine. If I could just say one thing in closing, you know, I grew up in Alice, and I was always interested in, you know, understanding how things worked, and you know, and I got into science, and I've had not only success at that, but I've had a really good time along the way. I think science can be fun. It's hard work. I mean, you have to have dedication and discipline to do it, but it can be rewarding, and it can be a career, you know, if you're so inclined. And if you are, just pursue your dreams, you know, pursue your commitment, and don't let anything get in the way. You know, you can find opportunities to allow you to do it, you know, not just during your education, but after that. So I encourage you all to, you know, follow your dreams, whatever they are.
Well, I'm glad you could make this presentation, sir. And I think on behalf of all of us, we are truly inspired by your work, and this this explanation of how you dove into it and experimentation and everything that went along with it was really enlightening. And I think this would really inspire us to pursue research in the future.
Good. I hope you do. So I was in a club like yours one time.
Okay, well then with that, I think we'll end the meeting then. Thank you, and have a great day.
Thank you guys for giving me this opportunity. I wish you all good luck, and just be safe, be healthy.
Thank you. Bye, sir.
Thank you.